Um, every single day, like tens of billions of your cells die.
Yeah, it's an absolute slaughter in there.
Right. Just today, like right now, while you are listening to this deep dive, there's this massive microscopic slaughter happening inside your own tissues. And and here is the paradox that completely blew my mind. Your survival depends entirely on them doing so.
Oh, totally. I mean, it is the ultimate biological sacrifice. If the cells inside your body suddenly, stop dying, you would be in serious life-threatening trouble very, very quickly.
Which is crazy to think about. So we're looking at a stack of pathophysiology research today that, well, it completely flips the script on how we think about death.
It really does.
Our mission for this deep dive is to look at the two main ways that cells die, which are apoptosis and necrosis. We're going to unpack the massive mechanical difference between a messy cellular death and a highly programmed one.
Yeah, and if we do this right, we'll actually discover why the classic textbook definitions of these processes are, secretly out of date. Exactly. Okay, let's unpack this. Because for the longest time, science treated cell death as a pretty simple, like, either-or situation.
Right. It was treated as a strict binary for, gosh, over a century. And if there is one single concept to kind of anchor you as we explore this, the cleanest summary from our sources is this. Apoptosis is a controlled demolition, and necrosis is a building collapse.
A building collapse.
Wow. Okay.
Yeah.
And understanding the mechanical difference between those two events, it's the fundamental key to understanding everything from, like, human development to how we diagnose heart attacks and even treat cancer.
So if we start with the building collapse, the messy one, necrosis, the term itself is actually ancient, right? It comes from the Greek word nekros, meaning dead body.
Yep, exactly.
Which, is where we get words like necropolis. But how did we actually start applying this to biology?
So if we look at the historical context, the German pathologist Rudolf Virchow really cemented this vocabulary back in his 1858 lectures on cellular pathology.
Right, 1858. Yeah, and he popularized this really famous dictum, omnis cellula, which means every cell from a cell.
Okay.
And this was a monumental push to describe disease at the microscopic level. But in that era, necrosis just became the descriptive term for dead tissue. Importantly, it wasn't seen as an active thing the cell was doing. It was viewed purely as a passive response to brute force injury.
And by brute force, you mean things like extreme heat or toxins, physical trauma, or like a sudden loss of blood flow, right?
Exactly, just pure external damage.
The best way I can picture necrosis mechanistically is like a water balloon popping. The cell sustains this overwhelming injury. It physically swells up with water. The outer membrane it can't take the pressure, and it violently ruptures.
Yep, that's exactly it.
And then all of its internal contents just spill out into the surrounding tissue.
And that spillage is exactly the problem. It's why it's a building collapse, because when that water balloon pops, it doesn't just release water. It releases digestive enzymes, ions, fragments of DNA, and very specific proteins. Yeah, it is. For example, there's this protein called HNGB1. Normally, this protein is like tightly bound to the DNA inside the nucleus of a healthy cell. It's locked away.
Right.
But when a cell undergoes necrosis and bursts, that HMGB1 gets thrown out into the extracellular space.
Wait, so things that are supposed to be locked deep inside the nucleus are just suddenly floating around freely in the tissue?
Yeah.
I mean, that has to trigger some sort of alarm.
Oh, it triggers a massive alarm. Your local immune cells are constantly patrolling your tissues. When they detect HMGB1 and, other internal debris floating around, they instantly recognize it as a severe danger signal.
Like a siren going off.
Exactly. They mount this rapid, aggressive response to contain whatever caused the damage. And this is why necrotic tissue is red, swollen, and painful. The bursting cell triggers a massive inflammatory fire.
Wow. So if necrosis is this catastrophic inflammatory mess that damages everything around it, our bodies couldn't possibly survive if that happened every time one of our billions of cells needed to turn over.
No, you'd be in constant systemic inflammation.
Right. So there had to be a stealthier way for a cell to bow out.
Precisely. And that stealthy way was missing from the textbooks for a surprisingly long time.
Really?
Yeah. Pathologists had spent a century cataloging how cells die from injury, but they totally lacked the vocabulary to describe cells dying on purpose, as a routine, healthy part of tissue turnover.
That's wild.
It wasn't until 1972 that three pathologists, John Kerr, Andrew Whaley and Alistair Curry finally coined a term for this process.
Yeah, I saw that in the sources. They originally called it something incredibly clunky, right? Like shrinkage necrosis.
Shrinkage necrosis, yeah.
Terrible name. But then they consulted a Greek professor who suggested apoptosis.
Right. Professor James Cormack at the University of Aberdeen, he suggested apoptosis because in Greek it refers to the falling off of leaves from a tree in autumn, or like petals from a flower.
It is so poetic.
It really is. It perfectly implies this natural, seasonal, pre-programmed shedding. It's a beautiful metaphor for the biology.
It is. Though I do have to point out a bit of trivia. Technically, the second P is supposed to be silent, like apoptosis. But, I mean, pretty much every biologist today pronounces it apoptosis, so we're just going to stick with that.
Yeah, I've never heard anyone actually say apoptosis in a lab.
Good to know. But I want to get into the actual mechanics of this falling leaf, because it is the exact opposite of the water balloon popping. It's a highly active, genetically encoded process. Instead of swelling up, the cell actively dismantles itself.
Right, it does. The cell quietly packs up its own infrastructure. The DNA inside the nucleus, the chromatin, is tightly bundled up and chopped into neat, manageable pieces.
Very organized.
Extremely organized. The cell physically shrinks, and then instead of popping, it actually buds off into these tidy little membrane-bound packages called apoptotic bodies.
Okay, so the outer plasma membrane never actually breaks.
Yep.
The water balloon never pops. It just pinches off into smaller, perfectly sealed mini balloons.
Exactly. Nothing spills. And because nothing spills, there is no HMGB1 leaking out, no danger signal, and therefore zero inflammation. It is entirely invisible to the naked eye. The surrounding tissue doesn't even know it happened.
I need to push back on something here, though, because when I first read about this, it felt like a logical trap.
Oh, how so?
Well, if apoptosis is this gentle, quiet fading away, like a leaf falling, and necrosis is this violent, explosive rupture, wouldn't apoptosis be the passive, low-energy way to go? It sounds like the cell is just taking the path of least resistance.
That is one of the most common and counterintuitive misconceptions in this entire field. What's fascinating here is that apoptosis costs a massive amount of ATP, which is the cellular currency for energy.
Wait, really?
Yes, the gentle death is actually the highly expensive one.
But wait, why does it cost so much energy to die?
Because it is a highly coordinated construction project. To execute its own programmed death, the cell has to actively spend its energy reserves to assemble complex protein machines called apoptosomes. Yeah, and it has to activate specific enzyme cascades called caspases, which act like tiny molecular scissors to carefully dismantle the cell from the inside out. Then it has to meticulously package itself into those tidy, membrane-bound bodies. It's incredibly hard work. Necrosis, by contrast, is often just death by energy failure.
Okay, let me try following this. Are you saying the explosive building collapse happens because the cell literally runs out of gas?
Yes. When a cell undergoes severe ischemia, meaning, its blood supply is cut off and it is starved of oxygen, it can't generate ATP anymore.
Right.
And without ATP, it doesn't have the fuel to run the microscopic pumps that keep its membrane stable. Water rushes in through osmosis, the membrane fails, and it pops.
That makes so much sense. The cell literally runs out of the budget to pay the demolition crew, so the building just randomly collapses.
It gets even more dramatic. In a laboratory setting, if you take a cell that has already initiated the clean, programmed pathway of apoptosis, and you intentionally poison its mitochondria halfway through the process so it loses all its ATP, switches tracks, it can't finish the controlled demolition, so it defaults to messy necrosis instead.
That is incredible. It really proves that apoptosis is a hard-coded program running on an energy budget.
It really does.
And speaking of hard-coded programs, there was a crazy statistic in the sources about a tiny nematode worm where the cells are like perfectly counted every single time.
Ah, yeah, C. elegans. This is a legendary model organism in developmental biology. So this little transparent worm develops from a single fertilized egg through exactly 1,090 somatic cell divisions.
Okay.
What researchers discovered is that out of those divisions, precisely 131 cells die by apoptosis every single time.
Wait, it's not like around 130 cells.
No, exactly, 131. In the exact same lineage positions in every single worm.
That level of biological math is staggering.
And this reproducibility was revolutionary because it proved that cell death wasn't just incidental wear and tear. By studying those 131 cells, researchers identified the specific genetic switches responsible for this programmed death.
And didn't that win a Nobel Prize?
It did. In 2002, Brenner, Horvitz, and Sulston won the Nobel for identifying genes like SED3, SED4, and SED9. They found genes that act as the executioners and genes that act as the protectors. And these turned out to have direct human equivalence, which fundamentally changed how we view biology.
And if you look at your own body right now, you can see the results of that exact same genetic program.
Absolutely.
Because around week 6 to 8, when you were in the womb, your hands started out looking like solid little paddles. There were no fingers. The only reason you have separate fingers right now to, hold your phone or steer your car is because the cells in the webbing between your developing fingers actively committed suicide.
It literally carves your fingers from the webbing, cell by cell, through apoptosis. And if that apoptosis program fails or gets interrupted by a genetic glitch, a child is born with syndactyly, which is webbed fingers or toes. It's a highly visual consequence of a failure to die.
But on the flip side, we can also see the physical reality of necrosis in modern medicine. Like when someone comes into the ER with a suspected heart attack, the doctors are dealing with necrosis, right?
Yes, myocardial infarction is the most visceral example of necrosis. If a blood clot blocks a coronary artery, the downstream heart muscle is starved of oxygen.
And so it runs out of ATP.
Exactly. Within about 20 to 40 minutes, those cardiac cells completely exhaust their ATP reserves and undergo coagulative necrosis. The cell pops, and all those internal muscle proteins and enzymes leak out into the surrounding tissue and eventually into the bloodstream.
Wait, so when a doctor orders a blood panel for a suspected heart attack, they aren't looking for a generic heartbeat signal. They're literally looking for the toxic waste spilled by popped necrotic cells.
Yes, exactly. Heart cells contain a specific structural protein called troponin. Normally, troponin is locked inside the intact cell, helping the muscle contract.
Right.
But when necrosis happens and the membrane ruptures, troponin floods into the blood. If you have elevated troponin in your lab results, it is a direct, measurable signature that the building collapse is happening in your heart tissue in real time.
And there's no equivalent blood test for apoptosis because if a cell dies quietly, it packages everything up, the membrane stays sealed, nothing leaks.
Precisely. The mechanical difference between these two types of death dictates how we actually diagnose a heart attack today.
So knowing all this, it's very tempting to look at the system and say, okay, apoptosis is the good, clean cell death, and necrosis is the bad, inflammatory cell death.
It is tempting, but we really have to abandon that framing. Necrosis is just tissue damage from an accident or injury. It's not a moral category, It's just a mechanical outcome. And apoptosis can be highly pathological.
Oh, so
Well, if your body triggers too much apoptosis, you get severe progressive tissue loss. This is the primary driver of neurodegenerative diseases like Parkinson's and Alzheimer's. It is the accelerated, inappropriate, programmed suicide of your neurons. Also in HIV, you see a ton of bystander apoptosis of healthy immune cells.
here's where it gets really interesting, though. What happens when your body triggers too little apoptosis? When the cells just refuse to die?
That is when you get cancer.
And the sources detail this massive paradigm shift in the mid-1980s regarding a gene called BCL2.
Yes, a huge turning point.
Researchers were studying follicular lymphoma, which is a type of blood cancer, and they found this gene was hyperactive. For years, everyone assumed it was a classic oncogene, meaning they thought it was a gene that, stepped on the gas pedal, driving the cancer cells to multiply and divide faster and faster out of control.
But when they finally mapped the mechanism of BCL2, it sent shockwaves through the oncology world. It wasn't making the cells divide faster at all. BCL2 is an apoptosis-blocking gene.
Wow. So if apoptosis is a controlled demolition, BCL2 is like a pair of wire cutters.
Yes.
The cancer cells were actively snipping the wires to their own internal dynamite so the demolition couldn't happen.
That is a perfect analogy. The cancer cells were simply refusing to die on schedule. If we connect this to the bigger picture, this discovery fundamentally changed our entire conceptual framework for oncology.
How so?
Well, a tumor isn't always a problem of excessive rapid growth. Sometimes it is purely a problem of a failed demolition program.
And knowing the mechanism actually allows us to fight it, right? Because modern therapies, like the drug venetoclax used for certain leukemias, they don't just act like traditional chemotherapy that poisons everything. They specifically target and disable the BCL2 wire cutters.
Exactly. They essentially restore the wiring, reminding the cancer cell how to pull its own demolition switch.
It's an incredibly elegant approach, but there is another dark side to apoptosis, even when the cell executes the program perfectly. We need to talk about the cleanup crew.
Yes, efferocytosis.
Right. When a cell breaks itself down into those tiny little apoptotic bodies, they don't just vanish into thin air. They have to be physically eaten and removed by patrolling immune cells called macrophages.
And the dying cell actually helps this process along.
Really? How does a macrophage know what to eat if there's no inflammatory alarm bell?
It all comes down to specific molecular flags. Normally, there is a fat molecule called phosphatidylserine that is kept strictly on the inner lining of the cell membrane. It faces inward.
Yeah.
But when apoptosis starts, the cell intentionally flips that fat molecule to the outside of the membrane, like raising a flag. Passing macrophages recognize that specific outward-facing flag as a highly specific find me and eat me signal. They engulf the package silently.
But what if the macrophages get overwhelmed? What if the cleanup crew gets delayed and those packages are just left sitting there?
This raises an important question, and it brings us to immunologist Polly Matzinger's danger model of immunity. If macrophages fail to clear those apoptotic bodies quickly enough, they don't just sit there perfectly sealed forever.
Right, they can't last indefinitely.
Exactly. Over time, those abandoned little membrane packages begin to degrade, they weaken, and eventually they rupture on their own.
Like bagging up your trash perfectly, but then leaving it on the curb for 3 weeks until the plastic degrades and it spills everywhere anyway?
That's a great way to put it. The scientific term for this is secondary necrosis. And when those bags finally rupture, they spill their nuclear contents, like that HMGB1 protein we talked about earlier, into the tissue. Suddenly, you have a massive danger signal and rampant inflammation anyway.
Oh.
In fact, this defective clearance, this failure to take out the trash, is now a leading hypothesis for the mechanism behind autoimmune diseases like systemic lupus erythematosus.
So the old rule we learned in biology class, apoptosis never causes inflammation, is actually wrong. The real rule is, efficiently cleared, apoptosis doesn't cause inflammation.
A very crucial distinction.
Okay, so we've mapped out this perfect two-part system. We have the clean programmed death and the messy accidental death. But the most recent papers in this stack basically say everything we just talked about is completely outdated.
Yeah, science moves fast.
If you only look at apoptosis and necrosis as this tidy dichotomy, you are about 15 years behind the science.
The textbooks had to be aggressively rewritten starting in the early 2000s. Pathologists realized they had a massive mechanical problem on their hands.
Because they were looking at cells under the microscope that look exactly like messy, unregulated necrosis. You know, the cells were swelling up with water, the membranes were rupturing, and there was massive inflammation.
But when they looked at the molecular level, they realized it wasn't an accident caused by brute force injury or a lack of oxygen. This explosive death was being actively triggered by specific hard-coded genetic signaling proteins inside the cell. Things like RIPK1 and MLKL.
Wait, so the cell was intentionally choosing to pop its own water balloon. Why on earth would a cell spend energy to trigger a catastrophic building collapse?
Well, think about it from the perspective of an incredibly clever virus. Many viruses have evolved to block apoptosis. Once they infect a cell, they snip the wires to the controlled demolition so they can keep using the cell as a factory to replicate.
Sneaky.
Right. But if the cell realizes it's infected and its apoptosis program has been hijacked, it needs a backup plan. So it intentionally triggers its own explosive rupture.
Because bursting open sounds the massive inflammatory alarm bell, warning the rest of the immune system that there is a viral invader.
Exactly. They named this backup pathway Necroptosis. It's a completely regulated death program that just happens to look like necrosis. And this discovery completely shattered the old paradigm. It proved that necrosis is merely a morphological description. It just describes what the cell looks like when it dies. It does not automatically tell you the mechanism of why or how it died.
That is a massive distinction. And understanding necroptosis isn't just trivia. It has huge clinical implications today, especially in the ER.
Enormous implications. Necroptosis is deeply implicated in something called ischemia reperfusion injury.
Ischemia reperfusion. So that's the damage that happens when blood flow is actually restored after a stroke or a heart attack.
Right. It's the biological equivalent of a flash flood. The tissue was completely starved of oxygen during the heart attack. But when the surgeons clear the blockage and blood suddenly rushes back in, the sheer mechanical shock and oxidative stress of that sudden oxygen delivery causes the struggling cells to panic.
Oh, no.
Yeah, they pull the necroptosis switch, intentionally exploding and making the tissue injury much worse. Finding drugs that can pharmacologically block that specific necroptosis pathway during those critical hours after a heart attack is one of the most intensely active areas of medical research right now.
And the list of death pathways doesn't even stop there. The Nomenclature Committee on Cell Death, which is literally an international group that categorizes how cells die, has to update their classifications constantly because we keep finding new, highly specific demolition programs.
The field is exploding with nuance, literally.
We've got pyroptosis, which is driven by an enzyme called caspase-1. It's not just a subtle death, it is a deliberate inflammatory alarm fire used by immune cells when they encounter certain bacteria.
Yeah.
And then there's ferroptosis, which is only described in 2012.
Ferroptosis is fascinating. It is an iron-dependent form of death. To use an analogy, it's essentially the cell rusting to death. The iron inside the cell interacts with the fats in the cell's outer membrane, causing them to oxidize and break down a process called lipid peroxidation. The membrane literally rusts apart until the cell dies.
It just shows that the clean two-category system we all learn in introductory biology is a massive oversimplification of a very messy reality. But before we wrap up, I want to do a quick misconception check on one more term that often gets thrown into this mix: autophagy.
Oh, yes.
It literally translates to self-eating. Is that another programmed way cells kill themselves?
I am so glad you brought that up, because it is a very common point of confusion. Historically, it was sometimes mistakenly called programmed cell death type 2. But the current firm scientific consensus is that autophagy is almost always a survival mechanism.
So it's not a death program at all.
No, it is a recycling program. When a cell is stressed or starving, it starts eating its own damaged proteins and old organelles, breaking them down into raw materials so it can survive the famine. It only contributes to the cell's death in very rare extreme contexts where it literally consumes too much of itself. So, we should not lump it in with apoptosis and necroptosis. Self-eating is for survival.
Okay, so what does this all mean? If we look at the journey we've taken today, we started in the 19th century with Virchow, just looking at dead tissue and calling it necrosis.
Yeah.
Then in 1972, we discovered the precise falling leaves of apoptosis. We learned that a gentle, quiet death actually requires a strict, expensive energy budget of ATP, while a violent, explosive rupture is often just a cell running out of gas.
Yeah, and we saw how that programmed death actively carves our fingers in the womb, and how the failure of the BCL2 wire cutters is a fundamental hallmark of how cancer survives.
And finally, we saw that the clean binary of messy versus programmed is actually a rapidly expanding spectrum, with things like the viral alarm of necroptosis and the rusting membrane of ferroptosis totally rewriting the medical textbooks.
And again, the crucial takeaway for you listening is that these pathways are not abstract biology. They are the exact mechanical basis for how modern clinics diagnose a heart attack from your troponin levels, and how the newest therapies treat leukemias by removing the brakes on cell death.
It is the microscopic battleground of modern medicine. As we wrap up this deep dive, there's one final concept from the sources that I want to leave you with, and it has to do with how we age. It's a concept called senescence.
Oh.
Sometimes cells sustain heavy damage, but for whatever reason, they don't undergo apoptosis and they don't pop from necrosis. They just permanently stop dividing.
Yes.
They sit there in your tissue, refusing to die, constantly secreting these low-level inflammatory danger signals.
They are essentially inflammatory zombie cells, and as they accumulate over decades, they are considered a major central driver of the aging process itself, degrading the tissues around them.
So here is a thought for you to mull over today. If senescence is basically a state where damaged cells refuse to undergo apoptosis and just linger like inflammatory zombies, what if the ultimate key to anti-aging isn't figuring out how to keep our cells alive longer?
Wow.
What if the secret to youth is actually figuring out how to remind these forgotten aging cells how to finally pull their own controlled demolition switch? Thank you so much for joining us on this deep dive into the fascinating microscopic universe inside your own tissue. Keep questioning and keep exploring.